And I think it does. So I'm going to show you guys a little video. By the way, if you guys are interested in EMS stuff at all, this is a really great channel. But how to do gloves. And let's see here. Let's do sound. Why not?
All right, guys, in the next 60 seconds, I'm going to teach you guys how to remove gloves safely and to represent any kind of contamination. I put some shaving cream on your gloves, okay? But before we get started, Mike, why don't you go ahead and introduce yourself and tell the class what you did?
Hi, I'm Mike, and I tried to use my mouth to remove my glove.
Yep, that's what we call a complete contamination. All right, guys, first thing we're going to do is we're going to grab the center of the first glove and we're going to pull it off. Good. No contamination. Next thing we're going to do is we're going to take our finger, we're going to stick it underneath the second glove, pulling up, and there you go. Zero contamination. You're welcome. Excellent job, everybody. Now throw your gloves in the proper container, wash your hands, and resist all want to yeet those gloves into someone's face. Wash your hands and get out.
I know what happened. It felt so natural.
Did I fail?
Okay, so there you guys go. No, don't do the next one. Okay. All right, so that is that. All right, now, slightly more serious stuff we're going to talk about, what we're going to be doing next week, which is microscopy and basic handling and culture techniques. It's basically two labs wrapped up into one, but they're going to overlap a little bit. Also, in terms of lab lectures, I typically have something called Cool Science Thing of the Week, which is at the beginning of each lab lecture, and it's the main focus for your bonus points on your lab practical. So each bonus point is worth 1 point, and usually the questions range about 70 to 80 questions. So that's a pretty good chunk of your grade. Like you can turn a B into an A if you get all of them right. So, but they tend to be a little bit harder because they're bonus points. So we're going to talk about this thing, which is this guy right here who does this really cool artwork with luminescent bacteria. Now, he makes all sorts of different types of artwork, and he has these Christmas ornaments that he was using, and he used this bacteria called Photobacterium phosphorium, and there's a couple other names too that might be more current, but basically because it's a ocean-dwelling bacteria, you constantly have to have a very specific type of media to grow it in. It needs to replicate the seawater environment, and you also need to constantly supplement it with 1% glycerol, otherwise the glowing stops, which isn't very good for self-contained ornaments. So we wanted to look at some other bioluminescent bacteria, and one of them he looked at was these species called Photorhabdus asymbiotica and luminescens. Now these ones are actually really interesting. They actually inhabit the guts of nematodes, which then inhabit the guts of large, caterpillars and stuff for certain types of moths and make them glow. Why do they make them glow? We don't know, but we do know that they glow. And so what he ended up doing was he took some basic media, grew it on there, and boom, you got some nice glowing. Great, that's exactly what he wants to see. So then the rule of thumb is anytime you have luminescence, you have to supplement it with some sort of extra nutrient source to make it glow more intensely. So he started adding in different things. He supplemented it with extra glycerol and extra glucose. And what we actually noticed is from the normal plate, it actually knocked down the luminescence for those two bacteria, which was weird and he had never seen before. He also noticed that this one then started producing a secondary dye with the increased supplementation, and he had never seen that before, which was really neat. This was the first time he was using Photorhabdus. So you can see how small changes in media really affect the growth characteristics of bacteria, and you can get some really big changes here. Now, why is this bacteria relevant to you guys? Well, it's unlikely that you'll see it, but you might actually see an infection with somebody with a glowing wound. We've seen this in history through different wars, World War I, and also during the Battle of Shiloh in the Civil War. We noticed that there were soldiers that had wounds that were infected, but glowing slightly, faintly at night. And what they found out was that those soldiers tended to have better outcomes for their wounds. And the reason why is because the Photorhabdus bacteria are really good at producing antimicrobial agents that kill off the other bacteria. They're not very good at infecting you, and your immune system just runs right over the top of them for the most part. And so the soldiers that had the wounds that glowed tend to do a lot better. So they called it angel's glow. And we also know that some adults will get this wound occasionally, typically people that work in dirt and soil, so like gardeners. And if you have a compromised immune system, so maybe you're elderly, one example of asymbiotica infecting an elderly person, he had a small glowing wound. It was very unusual, and he was an avid gardener. So you might come in contact with the nematodes or the caterpillars or just the bacteria that might be either of those two things right there. So you can see small changes, small little variations have really big impact on the way bacteria grow. So that's our cool science thing of the week. Next up, we're going to talk about microscopy lab. So safety stuff, we'll tend to have safety stuff at the beginning of each lab. And we'll just kind of talk about the important ones. Biggest thing here, make sure you guys always handle a microscope with 2 hands. They are kind of heavy. They're also really expensive and easy to drop because there's so many people in the lab. It's easy to bump into people. Always carry it with 2 hands. You've actually got a handle on the back and you just hold it on the bottom, and that's the proper way to transport it. Microscopes always go with the number assigned to your drawer. So if you guys opened up your center drawers when you were in lab where you were sitting, there's a number inside there. That number corresponds to your microscope as well. So make sure you grab that microscope and put it back in the same spot that you guys got it from. Carefully handle microscope slides. They are made of glass, and glass does cut if you break it and shatter it, so please be careful with that. Clean microscope lenses between slides and after you're done. So there's a stuff called optical lens white paper that's in your center drawers. You want to use that. You just tear up a little tiny piece, not a whole sheet, tiny piece, because lenses are small, and you just clean off the lens with that. For the slides themselves, you can actually clean them off with Kim wipes. It doesn't matter because you can't really hurt a slide. But you can just clean them off with Kim wipes so that they get the oil off of them. Last one, most important, only use the fine adjustment knob on the 40x and 100x lenses. The coarse adjustment knob will move the stage up and down quite a bit. And if you move it up too far, you can actually crack the slide or the lens on both of those objectives, and that's not a good thing. So please be careful of that as well. All right, so what will we be doing? Well, we'll be splitting this up into 2 days. So day 1, we're gonna have you guys use these microscopes. This is our current microscope setup. And day 1, we're gonna have you guys look at blood smears. These are human blood smears, and we want you guys to get familiar with focusing the microscope and hunting around the field for different types of white blood cells and red blood cells. Now you might have to hunt around for some of the white blood cells because they're not as common as red blood cells, which are the most proliferous cell types. But you will basically find them eventually, so you kind of have to hunt around and use the mechanical stage to move around the field of view to find them. Once you get done with that, we'll move on to something else in the lab, but for day 2, we'll come back to microscopes. In this case, we'll be using prepared gram-stained bacteria, and we'll be focusing on bacteria. Now, their bacteria are like, I don't know, 100 times smaller than human cells, depending on which human cell, and depending on which bacteria. But generally speaking, bacteria are much, much, much smaller than human cells. And so we will be looking at bacteria, because obviously that's the point of this whole course. So we'll be doing that. Now, we'll talk a little bit about blood smears and how to focus on them in just a second, but that's generally what you should be looking at. And this is the most important aspect. If you don't see nice, healthy, donut-shaped blood cells, then you're in an area where the blood smear is too thick or corroded. So when you do a blood smear, it tends to have a small area where it gets kind of shredded and clumped up and doesn't look right. And then you have like a nice Goldilocks zone, and then you get more terrible, terrible zones where it's all shredded and terrible. So you want to try to find that Goldilocks zone on your smear. Some of the smears are really nice and mostly Goldilocks zone. Other smears are not so much. So just keep that in mind. Okay, so when you sit down on a microscope, one of the first things you're going to have to do is you're going to have to adjust the oculars. The interpupillary distance for every human is different. And because these are shared microscopes, you guys are going to have to adjust it for your own eyes pretty much each time. What you want to do is you want to have your eyes relatively close to the microscope. That don't like have your eye sockets on the black part, but a couple millimeters away from it, not a huge distance away from the black part. And then you're going to pull it back and forth until it makes one large image. So if you're seeing 2 separate images or it's kind of like cut off and doesn't look right, you need to adjust that. And once you adjust it, you should get one single large circle. It takes a little bit of practice, especially if you haven't used a microscope a lot before, partly because your head tends to drift a little bit back and forth and to the left and right. So it takes a little bit of practice, but once you kind of get it down, you'll be able to do it much faster. And again, that's the whole point of today. So what you'll start out with is you will make sure that your slide is on the tray. You're going to clip it into place. You want to make sure that the microscope is set to the 10x or the 4x. I think all the microscopes have 4x. You can skip 4x if you want to, but if you don't feel super confident, you can start on 4x, or if you're confident, go to 10x. And you'll make sure the stage is all the way down, as far down as it can go. You clip the slide in, and you will start turning the course adjustment knob away from yourself, which brings the stage up. And as you keep going, you're going to be looking through the microscope to find a point in which the bacteria start coming into focus, or the slide of blood cells. So basically, you're looking for it to get kind of better. You won't see anything for a while, and then all of a sudden you'll see some fuzzy stuff, and then all of a sudden it'll come into sharp focus. Once you're done kind of getting it into rough focus with the course adjustment knob, you will then use the fine adjustment knob to dial it in. Now, cells, even though they're very small, are still technically three-dimensional, and so you can kind of be in focus and out of focus, above or below, or right in the middle where it's nice, depending on the object that you're looking at. So you kind of have to adjust it for each object, but sometimes you can overshoot it and go past the nice focus into the bad focus again. So just keep that in mind. So here's an example of a cyanobacterium, which is very three-dimensional, and you can see this is above it, and you can see everything is very fuzzy right here, but as you increase focus, you see the majority of this in focus. It's still a little bit fuzzy, but if you go past this, now you've got certain parts that are in very good focus, but the rest of it's now fuzzy. So that's the kind of thing that you have to be careful of, just dialing it in perfect for your needs. Now, important to note, don't touch the course adjustment now again. Just leave it alone. You won't need it for the rest of that time because you're going to crack the lens if you use it on the 40x or the 100x lens. Okay, so you'll then rotate the nose piece over eventually to the 40x lens. And when you do that, one of the things that's going to happen is if you're in focus on the 10x lens, so the total magnification is actually 100x because the oculars have their own power. If you're in focus here and you click over to the 400x, you should still be in focus when you go over the 400x. Because of the way the microscope is set up, they're parafocal. So once you click it over, it should be roughly in focus. You might have to do a little adjustment, not too much, it'd be good. But if you're a little out of focus over here, when you click over, sometimes you'll be in this range where you're out of focus. So don't panic if you can't see anything. What you'll do is you'll do a quarter turn forwards, a quarter turn backwards, whichever way is better. Keep going in that direction. If neither is better, go to a half turn, all the way up to a full turn on the fine adjustment knob. If neither is better, then start back on 10 and work your way up again. Or ask TA for help, because that's what they're there for. After that, once you get into focus on the 40x, you're going to then rotate the nose piece again to the 100x. But before you do that, you're going to click it actually in between those two, and you're going to add a little dropper of oil. The dropper of oil is in these white trays on your table, and they got little blue caps on them. You just put one drop of oil on there, and then you'll rotate the 100x into place. Now, same kind of thing here. If you're not perfectly dead in focus here, it's going to get really wildly out of focus on the 1000x. So just keep that in mind. And the reason why we have to do oil is because the light is so close to the light source that as it's shooting through, it's going to scatter heavily, and you'll be able to not be able to focus. So if you didn't put oil on, it's just going to look fuzzy and not very in focus ever. But if you put the oil on, the oil directs light so it goes straight back up and makes everything nice and crisp and clear. So if you're having a really difficult time focusing on the 1000x, check 1 if your slide has oil on it, and check 2 if your slide's upside down, because that's another issue. The thickness of the slide is so thick that you wouldn't be able to focus it on the 1000x, but you can focus it on the 40x or the 10x. So keep that in mind. Okay, any questions so far? Makes sense. Okay. So human blood smears, a couple of key features that you guys want to be looking at. First one is red blood cells, which are these guys right here. As the name implies, they are red and they are blood cells, or erythrocytes. Fun fact, they're one of the very few cell types in the human body that has no DNA in it. They are genomeless. Weird, right? Okay, the white blood cells, on the other hand, are not. They have interior DNA. And the reason why we know this is because we stain it with a nucleophilic DNA, nucleophilic stain. And so for each of these white blood cell types, they have different types of nucleuses, which are actually really easy for us to differentiate. The first one is neutrophils, which kind of has these multi-lobate nucleuses. So it's actually kind of really easy to tell because it kind of looks like chains of dumbbells. That's what it looks like to me, at least. And then kind of a reasonable amount of cytoplasm. The other one here is the monocyte. It's kind of got this weird bean-shaped nucleus with a bunch of cytoplasm. It is pretty easy to see. It's not quite as common as a neutrophil, but you will see it from time to time. Basophils here have bilobate nucleuses, so they have two little lobes right there, and they have these gritty purple granules. So they're really easy to differentiate because they've got grittiness inside of them that's purple. That's a basophil. Lymphocytes have a very large round nucleus and this tiny little crescent of cytoplasm like this. So people often confuse monocytes and lymphocytes, but lymphocytes can be differentiated easily because they're mostly nucleus and just a little cytoplasm. Monocytes have a lot of cytoplasm. Last one is eosinophils, which also have a bilobate nucleus, but they have gritty pink granules in them, and so they are easy to differentiate from the basophils. So they're both gritty on the inside, and they both have granules, but one's purple and one's pink. Now, key thing to note, everything on here is relatively common in blood except for basophil and eosinophil. They are very hard to find, much like Carmen Sandiego. I don't know if you guys remember Carmen Sandiego. Skit show a long time ago. Okay. Carmen Sandiego is very hard to find. She's a criminal. Okay. But these are not criminals, but they are very hard to find free-flowing in blood, in part because they tend to hang out in very specific tissues. So we don't see them in free blood as much. Something like 0.4% and 0.1% of blood is basophils and eosinophils, respectively. You don't have to know that. You just know that it's rare. Okay. Day 2 for microscopy, we will be looking at some bacterial slides. You'll have gram-positive bacteria, which are purple in color, and you'll have gram-negative bacteria, which are pink in color. You're going to look at both, and you're just going to figure out what kind of shape and arrangement they have. You're going to take a picture of them. You're going to just practice getting your microscopy skills in good standing. And you want to go up to 1000x for these ones. Pretty simple, pretty easy, fun little thing to do. Not that much stress for that one, okay? All right, now the one that's slightly more important and will be kind of the foundation of what we'll be doing for the rest of the semester is basic handling and culture techniques lab. A couple of safety things. If anything breaks in lab, make sure you let your instructor know right away and we'll clean it up together. We will be using BSL-1 organisms in lab, but we are going to treat them as BSL-2 just to maintain the same kind of safety procedures all the way through the semester. Once items are inoculated, they will go in the incubator for your lab section. All stock cultures will be disposed of in the proper locations. Your TAs will tell you guys that again during lab so you know where to put them. You'll disinfect your working area and your instruments prior to using them, as well as after you get done using them, so that we're not contaminating our work area after we're done. Make sure, of course, you wash your hands before and after lab so that their hands are nice and clean. Okay. So, basic handling and culture techniques. Day 1, pretty simple. We'll start by learning the foundational inoculation techniques and make cultures that we will use on day 2. And for day 2, we will actually be using a micropipetter, practicing that. I'm sure you guys have, probably most of you have used a micropipetter before. We're going to be using them in a little bit different manner because we want to enforce a sterile working environment. We'll talk a little bit more about that in a second. But then we'll use the culture that we made from the day 1. We'll look at them for cultural characteristics. And then day 2, we'll use them to help us make a wet mount slide so that you guys can view those cells on your microscope. We actually don't need to do any staining for these because of the way we're going to be doing it, but you guys will actually get a chance to do that. Okay, so subculturing. What you need to know. It is the process of transferring organisms from one media to another using aseptic technique. How do we do that? Well, we've got a number of instruments in your lab drawers. You guys should each have, in the center drawer, a loop and a needle. Most important thing, make sure you guys put that back when you're done. Don't put it in your neighbor's drawer because then it gets really confusing as to who has what and it makes it very difficult to operate the lab. So keep that in mind. We'll use a loop or a needle depending on what we're trying to accomplish. So there's certain rules for it. So if we're going to use a needle, we'd always want to transfer something from some type of media and we're going into a slant or a deep. The reason why we use a needle is because it creates a nice thin line, which is very important for slants and also very important for deeps because we have to poke into the auger for that. For inoculating loops, we'll always use loops for a streak plate. The reason why is needles are very pointy, and if you are trying to drag a needle across the surface of the auger like that, it will tend to gouge the auger and create gashes, and then it won't look right. So please make sure you guys are using the appropriate tool for that. Last one is for broth. It doesn't matter what we use. You can't hurt a broth. Use whatever one makes you happy. There you go. Okay. So first thing we're going to talk about really quick is streak plate technique. This is the one that's kind of the most challenging because it's skill-based. Basically, this is the way you isolate bacteria to get one single colony. If we say that there is one single colony that we tend to believe, how true that is debatable. But generally speaking, we say if we have one isolated colony, that was one bacteria that created that colony. This is very, very useful for when we have something like a mixed culture, which most bacteria in the wild are actually in a mixed culture. And so if we're trying to separate them out, we can say, okay, this one colony that's not touching anything else, that's just one genus and species of bacteria. And that's why we need to use this technique. It's very, very useful techniques. There's different ways to do it. So this is one example here. This is with a bacteria that's got a gene called P-glow that makes it luminesce very nicely. But we will instead be using this one, which is a little bit easier for us to use and doesn't require as much work. So I'll show you guys how to do that here in just a second. So we'll start out with a plate. When you guys have a plate, TSA plates always have two parts of them. They have a bottom and a lid. Whenever we label TSA plates, we always label the bottom. The reason why is because you can't take a bottom off and like swap it around, so you always know what's in the plate. But you can take a lid off and put it on another plate, and now you don't know what it is. So we always label the bottoms of plates. For this one, we'll draw a line down the center and a line across. And then whenever we inoculate anything in lab, we're always going to put our initials, what it is, and the date. This is a universal rule for that. If you get hit by a bus and you're like, hey, we don't know what this is in this lab, now we do because you put your initials and date, you labeled everything appropriately. Labeling is actually really important. Please don't get hit by a bus. But it is actually, you would actually be surprised how often scientists who were working on something very important and had their own special system of knowing where everything was ended up dying, and now they have a laboratory full of dangerous things or important research, and nobody knows what anything is. It actually happens quite a bit more than you'd think. So proper labeling, very important. Okay. So now you've labeled, you've got your plate, you've got your loop. First thing you're going to do is you're going to decontaminate the loop. And the way you do that is with the back incinerators that we have at your tables. There is a video on Lab Archives that shows you guys how to use them. Please go watch those videos. They're very handy, and it took me a lot of time to record all of them, so please do that. You'll take your loop, you'll stick it in the back incinerator. It only has to sit in there for a couple seconds. It's very hot in there. It's 450 degrees Celsius, I believe. So pretty hot. You'll take the loop out and you'll let it cool down for a second. You will take bacteria from your stock culture. And the way you do this is let's pretend you have a broth. So you hold it in your non-dominant hand. You have your loop in your dominant hand. You're going to take the lid off of the tube, stick the loop into the stock culture, swirl it around, grab some broth, take it out, recap it immediately, and put it down. Then you will take your plate and you will lift the lid of the plate. And for this one, you'll want to have your hand on this end of the plate and then lift up the lid just a little bit. You don't want to take the lid off. The reason why is because the bacteria can fall in from you just breathing or from the ceiling or the vents. So just lift the lid up a little bit. You'll take your loop and you'll stick it in here and point it towards your palm and gently rub it up and down in this pattern here, starting in the back all the way to the center line. You want to do nice long strokes. Once you get it all the way there, you will then close the lid, take your loop, and put it back in the back incinerator to decontaminate it. Then you'll take it out again. You'll turn this 90 degrees so that your hand is now right here. You'll lift it up, and you'll stick the loop in over here and drag across here. Now, we're not putting more bacteria on. We're actually just moving the bacteria that are right here over and trying to reduce their numbers. And the reason why is because we want to get that isolated colony. Right here, it might be too thick. We don't know because we don't know the concentration of bacteria. Right here, we might not get an isolated colony. But in the last one, which we will repeat this step again by turning it 90 degrees, flaming our loop, letting it cool down, and dragging it across again, we should definitely get an isolated colony. It works pretty well. It's not that hard. Most important thing out of this, I don't know, there's a lot of steps. This is a technique that's not really kind of talked about in the lab a little bit, but I'm just going to give you guys a heads up. If you go toward the top of these arches, there's actually a lot more bacteria there than you'd assume. You want to kind of drag through these center areas, so right here on these little lines, because this will give you the right amount of bacteria. If you drag up here and you like cross two of these lines that are the arches, you're actually dragging a whole bunch of bacteria. So don't do it on the arches, do it in the middle, in the center lines. You'll do all that, you'll flame your loop one last time, you can put it down now because now it's decontaminated. You'll tape your plate up and you guys will stick it in the incubator once all of your lab partners are done. You guys will grow it, and then you guys will analyze it for cultural characteristics. Now, aside from the plate, you guys will also get a TSA slant and a nutrient broth. Nutrient broth, very easy, very similar thing. Your TA is actually going to demo it for you. I know it's hard to visualize me holding these things in my hands because they're not here. But your TA will show you guys the proper techniques for holding these. But they are listed in your lab manual, so if you guys review that, you should be good to go. You guys will transfer an E. coli sample to a slant and a S. cerevisiae to a broth. You'll then make a streak plate of Serratia marcescens, which makes a nice red pigment, sometimes, depending on the temperature. And we're going to place all of our cultures in a rack for your group. And we'll put that rack in the incubator, and the plates will just be on their own, separate from the rack. Pretty simple for that. Now, the day 2 stuff, we're going to get into pipetting. Now, I know you guys all know how to use a pipette, more or less, but we're going to go over it again just to make sure. Most important thing from here is that there are 2 stops in a pipette. The first stop is just going to suck up the amount of liquid you need. That's exactly what is set on the indicator window. The second stop, if you go down from the first stop to expel the liquid, the second stop will blow out any remaining liquid from the tip. This is very useful for that, to kind of get everything out of there. And some pipettes, just so that you guys are aware, have a third stop which ejects the tip, which is never fun to accidentally eject your tip into whatever you're working on. But ours don't. Ours have a tip ejector button on the back here, which is very hard to press. So just keep that in mind. You won't accidentally eject it. Now, for this, you do need to kind of understand the process of setting volumes. Whatever the top number is the maximum number of this. So this one's listed as a P20, a P200, and a P1000. A P20 only goes up to 20 microliters. So you can scroll this all the way up so that this top number is going to be that tens position to a 2. The rest of these would have to be a 0 and a 0. We can't go past 20. So we'll set it 20. If we wanted to pipette 15 microliters, we'd set it like this: 1, 5, and 0. So this is the decimal. If we wanted to set it to 15.3, we just keep moving this over until it's got to the 1, 2, and 3. Okay. So that's how you set it for the P20. P200, very similar thing. The top number for this one would be the hundreds, the middle number is the tens, and the last number is the ones. And within this, there's a little bit of variation. So if you wanted to set it as like 75.5, you could do that kind of, but it's not the most accurate once you're getting to that lower number. And the last one's a P1000. This number is the very top. It would be 1. The hundreds would be in the second position, and the tens would be in this last position, and then ones would be these little tiny tick marks right here. There's an optimal range for each of these to be used in. So for example, both a P200 and a P1000 could pipette 200 microliters. But the P200 would be much more accurate because it's designed to pipette exactly 200. So its accuracy would be much more consistent than the P1000. Even though you could do it with the P1000, its optimal range is more in the upper range. So you could do it, but you'd want to use this one as the optimal one. So try to pick one that has the number kind of in the middle or at the max range of it, not at the very low end. So you could pipette 1 microliter with a P200, but I can tell you from experience, it's really, really hard to. So there you go. All right. So what you guys will be doing on day 2, you guys will be practicing pipetting liquid into separate microtubes in different volumes. Once you guys get kind of confident with that and feel that you've got it done appropriately, you will then look at your plates and tubes from day 1, observe the cultural characteristics, and then you guys will start doing your wet mount. For the wet mount, you guys will use 25 microliters from your Saccharomyces broth, so just regular old yeast, that you guys made. So make sure you guys do that properly. You will pipette 25 microliters onto a slide, and we will give you guys a stock culture of E. coli broth, and you'll pipette 25 microliters onto that slide as well. You will then take a sterile toothpick that we'll give you guys. You will do a scraping of the inside of your cheeks and rub it back and forth in that broth. This will deposit some cheek cells on there, and then you'll dispose of the toothpick properly. We'll have these bleach buckets on the table, so you just drop it in there. And then lastly, you guys will try to get a hair. It doesn't have to be a long hair, just a tiny hair, and you'll put it on the side, and then you'll cover it with a cover slip, and then you guys will look at it in the microscope. And what you should see is something like this. So in this, you can actually see the hair right here. And these large oval cells that you can see right here that are kind of very translucent, those would be the Saccharomyces, the yeast. This larger thing right here, this kind of dark colored thing right here, that's actually a cheek cell. So you can see several of the cheek cells right along here. They like to collect along the edge of this hair. I don't know why, but they do. And it's very, very difficult to see on camera, but if you guys look very closely, you guys will also be able to see the E. coli, and because E. coli is motile, it'll be kind of swimming around in the broth, and so you should actually be able to see that movement. It might be really difficult too, but your TA should be able to set one up on the instructor microscope because it's designed to do that part a little bit better, and so you guys will be able to see those, even if you can't see the E. coli. Okay, let's see here. Still good on time? Yes, we are. Okay. Now, and then once you guys are done with all that, you guys will take your slides and dispose of them properly, which we'll talk about in a minute. Most important things. One, make sure you guys get all of your safety forms and safety stuff taken care of. If you didn't have all of your safety stuff to take a picture of and submit it in lab archives, make sure you guys do that as soon as you can. If you guys weren't able to do it, still aren't able to submit it and get it all taken care of, Monday, as soon as you walk in the lab, you want to take care of that right away, or else your TA is going to kick you out. Make sure you guys do that. Safety form also needs to be signed. You can take care of that right now, so make sure you guys do that. There will be a pre-lab quiz. It'll be available in a couple hours. You guys will have until Monday morning at 8 a.m. to take it, so make sure you guys do that before Monday morning at 8 a.m. You guys have a safe weekend. You survived the first week. I'll see you guys next week, and swing by and see me if you have any questions.